All right. Hello, everyone. So I thought that I would try to record a couple of these, maybe some PowerPoint lectures, just to try and make things a little bit more interesting than just a pencil and paper type worksheet or something that you guys are doing. So I hope everybody's doing well. I know this has all been pretty crazy. I said that all in another video. So if you want to check that out, it's on the Google Classroom. But like I said, hope you're staying safe. Hope everything's going well. So one of the things that we're doing is we're going back and we're focusing on some older chapters. I'd like to do some stuff that we were planning on doing, but right now I'm just trying to keep things as simple as possible in many ways. And so let's go back through and look at a chapter that we had a lot of fun with, but that also had a lot of information. And I'd also like to do some labs with that as well. So let me share this screen with you here. Okay, so let's look at this and let's see, move that out of the way. Okay, so our census chapter, we spent a lot of time in this chapter, but I think it was also hopefully one of your favorites because there was a lot of information in it and a lot of it was really applicable. So my hope is too to record some videos. I went to school and kind of took a whole bunch of things from my cabinets, and some of those were some different dissections. I brought in a cow eyeball home. I'm going to try and talk my son into helping with that in the next few days. So hopefully you find that video as well. Obviously you can't do that one at home, but I'm hoping that the equilibrium lab, remember we kept that for a while and we were going to do that when it warmed up. Well, yesterday it was 86 degrees, so we can do these things in your backyard now, or maybe you can do it in the basement if you have a nice finished basement. Okay, so look for those things, but for right now, let's just go over some of these notes with the census. I don't want to read every single thing to you. These are all still in your notebooks. Some of you may have left your notebooks at school. It's on the wiki as well. I'm trying to make this to where we can access things as best as we can. So when we were looking at the eye, one of the things that we talked about was optical illusions. So if you're getting bored with some of this stuff that's going on, I know it's probably, if it's anything like me, you're kind of stressed one moment from different things going on, and the next moment you feel a little bit bored just because you're in the same place and the same people and all of those things. So maybe try to do something somewhat constructive. I'm sure you're probably spending a lot of time with TikToks and things like that. But optical illusions, there were a lot of different videos that we could have looked at. And so we looked at this one with the shiny legs or the white paint. We watched the Good Mythical Mornings. There were a couple of those. There were quite a few videos with those. And here's some of those links as well. Special senses, remember, were things like our smell, taste, sight, hearing, and equilibrium. Touch did not count as a special sense. And we spent a lot of time on each one of those. And they were either large complex sensory organs like our eyes. Remember, 70% of all of our nerve endings were in the eyes, or 70% of the brain focusing on the nerves. Or they were clusters of receptors like our taste buds and the olfactory. Okay, accessory structures were the things that were on the outside, like the external eye muscles. We had 6 of those, the eyelids and the lacrimal apparatus. Spend a little bit of time talking about these 2 types of glands. Meibomians were the lubricant, and the lacrimals were the ones that release tears. Remember, the lacrimal glands will release it, and then it goes across your eyes to help flush it to the nasolacrimal duct. I know many people were surprised that when you cry, that's one of the reasons why you get snot, is because those tears actually go in that duct and come down into your nose. Okay, the external muscles, we talked about that. There's one on the top, the bottom, and each side, and that's how it allows us to go look up, down, left and right. And then we also have these that will help the eye kind of rotate around. We dissected the cow eyeball. They didn't have those. They just had the 4 muscles. The 2 different types of humors were the aqueous humor and the vitreous humor. Okay, so the aqueous humor is the one that's right up here in the front. I remember aqueous is anterior, and the vitreous, when we dissected that cow eyeball, it all just flopped out. The 3 layers, don't want to spend a whole lot of time focusing on that, but the outermost is the sclera. That's the white part of the eye, also the cornea. And the middle is the lens and the iris. And that's where we also find the pupil. And the innermost is the retina that has those photoreceptors. We did that blind spot experiment, if you remember that, and you want to use that with your family members, if you're watching your little brother or sister. Remember the blind spot? We had the X on one side and then the circle on the other, and you stared at one with the eye closed and you zoomed in and you would have that little blind spot. That's where that field of vision comes right on the retina where the optic nerve attaches. And then of course we have the difference of the rods and the cones. These pictures are showing the tapetum that's in the back of the eye, or the tapetum, and that is that reflective part that is going to help try to, give a little bit more night vision to some animals. We don't have that. The cow eyeball did. Okay, so here's our diagram that we've looked at multiple times. Okay, spend a little time on that. That'll be on your lab sheet as well. The lens is what's actually causing us to focus. Okay, so the power of the cornea remains constant, but the lens can change. And so these special muscles that are on the lens are going to get larger or smaller, or they're going to pull the lens to be more flattened or more oval shaped. And that's what's going to focus the light in different situations. Okay, that's just what I explained. Accommodation is to focus on closer objects. And then of course, the brain is what corrects those objects and helps to interpret it so it sees it correctly. All right, moving on to the ear, trying to do this relatively quickly. Remember, our hearing is a wide range, okay? And these are called mechanoreceptors. They're picking up on mechanical movement. And what's really the mechanical movement is those sound waves are causing our eardrums to vibrate and to move. Okay, so we hear from about 20,000 hertz all the way down to 20 hertz. And this diagram shows our hearing range compared to lots of other ones. Bats have a very, very high frequency. That's why we can't hear all of their noises that they're making, but they can hear them. Okay, remember the three main areas. The external ear goes all the way to the tympanic membrane. The middle ear is this chamber right in here that's filled with air. Okay, and then the inner ear is over here. The outer and middle are just hearing, and the inner ear is hearing and equilibrium. This is a better picture. If you remember this, okay, this is the auditory tube, or also called the eustachian tube. And what this does is it's normally closed, but when you yawn or swallow, it opens to help equalize the air that's in the atmosphere to what's in that middle ear compartment. And so that's why when you go down into a deep valley, now there's more air pushing in from the outside because there's more pressure in the valley. And so when you yawn, it sucks in a little bit more air pressure through your eustachian tubes there and allows that to equalize. Same thing on the inverse is when you're going up in an airplane, now you have more pressure from this sea level air right here, and it's pushing out and bulging on the eardrum. And so then when you yawn, then that opens and equalizes the ear or the air. Okay, so pinna, external auditory canal. Talked about Q-tips and how we're not supposed to put them in the ears and things like that. Okay, sound waves enter and cause this to vibrate, and we have the ossicles, the hammer, the anvil, and the stirrup. Okay, those are the three smallest bones. And remember, we said sound travels a lot better through solids than it does through liquids or air. And so as a result, these are actually acting like amplifiers. They're going to amplify that sound to help travel it down to the inner ear. Spend a little time talking about the osseous labyrinth and the cochlea. Amplitude's going to increase. We just talked about that. And that's going to stimulate the brain through the cochlear nerve. And then finally, with the hearing part, we had equilibrium. Okay, so equilibrium is kind of difficult to describe. Everything works fine until you really feel like it's not. Like I can describe to you these pictures behind me and what my children look like, or I can describe to you looking outside and the green trees on the leaves, the leaves on the trees. But to say my equilibrium feels fantastic today, it's going to be a little bit different to describe. So you don't really know until something is wrong. These are in the vestibular apparatus and the rotational and the gravitational. Okay, so there's different ways to describe this. The rotational is when we get the rotating or angular movement, and then gravitational is the vertical and horizontal. Okay, and so what really happens, I kind of picture it like when you put the milk jug into the refrigerator, you can kind of feel it. It's resisting when you move. That milk is going to kind of stay in one spot when you grab it quickly. Okay, that's like this. These hair cells are in this fluid, and when you move, the fluid kind of flows over those hair cells and causes you to feel like it's moving. Okay, and then deafness, we spent a little time talking about that. Take care of your ears. And then finally, the chemical senses. We said it's much less than other animals. My kids and I were watching a show the other day about dogs, and I want to say that bloodhound dogs, which have the best hear or the best smell, was somewhere around 60 times what we could smell, which is crazy. And I'm sure there's different ways that other sources might describe that. Okay, so we can still smell better than any machine, and those olfactory receptors pick up on information that sends it to the brain, and we said it was closely linked to the limbic system. The limbic system is what's going to be part of our memory that's associated with emotions, which is why when you smell certain smells, it might remind you of a certain person or a certain time. There might be things that you smell now that when you smell 10 years from now might remind you of what we're going through right now. Okay, most of our deficits are result of olfactory. We talked a little bit about how we can adapt to that unchanging stimulus. And then finally, the last thing was about taste buds. The 5 basic ones were sweet, sour, bitter, salty, and umami. So if we did our taste lab with that, which was pretty fun, we tasted lots of different things. I might be able to do a little video with that, or a little assignment with that. That might be something fun that you can do with your family again. So remember, when we smell things and taste things differently, like that, the video we watched about the Smellovator, where they could smell one thing and taste something else, and how it tricked the brain. So anyway, that's the whole chapter in my hope 10 to 12 minutes. Hope you're doing well, and let me know if you got any questions, if I can help you out with anything, and we'll talk to you again soon.